I want to welcome my big sister is here. She can't talk, I don't think, because she had a pretty bad accident last week. But Nancy's here. Nancy's iPad. That's my big sister. That's Dr. Salini is actually here and recovering from her unfortunate accident last week with 3 breaks. So welcome, Nance. Welcome everybody else across the United States, Canada, Mexico, the Dominican Republic, Puerto Rico, Colombia, Spain, and of course our friends over in the United Kingdom and the Republic of Ireland case. So welcome everybody. Glad you're here. It's going to be an amazing Zoom-tacular day, to say the least.
John, as I say, we've still got people joining. I mean, once again, we're letting them in here one at a time as they come in, but we're adding about 5 or 6 people a second here, so it'll take a little more time. Mr. Molson, how are you today?
Fine. Is Dr. Linseman on?
Yes. Are you kidding? Absolutely.
Okay, good.
Absolutely, he's here.
That's all I care about.
Oh, look at that, John. He doesn't care about you or me. Hey, John, you like my background?
I love it. Now, is that your brain or it's John's brain?
Well, when you're in the room, the smartest man in the room is John. If you don't believe it, just ask him.
No comment, especially with Dr. Linseman on here today.
I know, but we've got to have a little bit of fun here. And once again, we've got a lot of people joining here, so we're going to give it, we don't want anybody to miss out because what they're going to hear. And once again, I can simply tell you, we got Dr. Jeffrey here, and I think I've unmuted him already. Dr. Jeffrey, are you there? We don't see your video though,
Yeah, I'm here.
Should if they've got somebody that they think they might want to get on, might want to hear what we've got to say, what do you tell them?
Listen.
Can't hear you, Dr. Jeffrey. Should they make that quick call and say, hey, make sure you click that link?
Absolutely. Get on the call, dial up the number and sit back and be ready to be blown away.
Okay. All right. We've got John. I'm still just clicking them and letting them in here. Once again, as everybody knows, after the way Zoom works right now, we've got to take a little bit of time and do a little bit of housekeeping here to let people join. But, it's going to be an exciting time here. I mean, I was very taken, Dr. Linseman, with what you shared with us yesterday and got on the phone and invited a number of people to be here. So we've still got people joining, John, but big numbers coming in here. So.
Well, we're giving another minute or two, but in fairness to the people who are on time, we should start here in about 60 seconds.
Yeah, we give for the ones. Once again, that's why we're doing that. Once again, these Zoom calls are for you guys. We set the bear trap up there in Montreal and got John Molson, in that bear trap so we can get him on these calls to share some incredible information and some incredible resources. And Dr. Linseman is certainly one of those incredible resources. So once again, make sure you show your appreciation to Mr. Molson. Good morning, Doctor. How are you, Dr. Linseman?
Good. How are you doing?
Well, I tell you what, yesterday, as I told my friends, I've been around Immunotech for 24 years, about 2 weeks after John Molson showed up. And yesterday was very enlightening with the information that we were going to share today. So I got on the phone and twisted some arms, to say the least, and some people may even say I was somewhat threatened. but they need to be here today. So, all right. Well, John, they're still coming in. We want to give it just about another 5 seconds. Let's give it 10, 15 more seconds, Doctor, if you don't mind, and because they need to hear what you have to say. I mean, with what we saw yesterday, it's just imperative that they don't miss out, and it's just not the same when they listen to the recording as being live. So,
yeah, Keith, it's John. It's a special privilege to have the investigator talk about it in his own words. So it's a very unique situation.
Absolutely, and thank you, Mr. Molson, for putting that together. I mean, without you, it wouldn't have happened. So, once again, thank you very, very much for that. All right, well, Mr. Solider, shall we?
I think so. I think it's time to go to work. Welcome, everybody, across, as I said earlier, the United States, Canada, Mexico, Colombia, the Dominican Republic, Puerto Rico, Colombia, Peru, Spain, the United Kingdom, the Republic of Ireland. I think we even have some people on from Ecuador today, Keith, that I talked to for a bit yesterday. So really, we are a worldwide enterprise. And, you know, for those of you who are just getting familiarized with Immunotech Research as a company, and I've been doing this, I'm an industry veteran. For those of you who don't know me, I've been in network marketing full-time since 1983. Some of you may not have been born then, but I started as a college student way back then, and I'm still doing it. I'm approaching my 60th birthday. So I've seen a lot in those 37 years. And I've never seen a company like Immunotech. And you say, well, you know, defend that statement for a minute. Well, here's how I would defend it. Our core product, Immunical, the research began in 1975 with a collaboration of Dr. Patricia Kongshaven and Dr. Gustavo Bunos. Dr. Kongshaven, as some of you know, joined us a few weeks ago on the Zoom-tacular for June. Still going strong as she is approaching her 90th year of life. Amazing woman, to say the least. But 1975, and just, you know, put that in context. Where were you in 1975? I was a freshman in high school. Some of you weren't born. Some of you a little older. Perhaps you were in college, or perhaps you were out working already. But 1978, the first actual scientific research paper written about Immunical was published. What happened in the ensuing years? Research took place at places like the Pasteur Institute in Paris with Dr. Luc Montagnier. Dr. Wolf Droge was doing some things up at the German Cancer Institute in Heidelberg, Germany with this product. Other facilities, researchers around the world were working with it. Finally, in 1996, after some 21 years of research, did Immunotech Research Company start as a commercial enterprise. Now think about that for a minute. 21 years of research done before this ever became a commercial enterprise. Now, one of the three people who actually decided to bring this product to the world is actually with us today. He's become a very good friend of both mine and Keith and many of yours on the call today. He's from the probably the most famous family there is in Canada. That would be the Molson family. And he guides our research. He works with researchers literally around the world, and he has for the past 2.5 decades and done an outstanding job. So I'm going to turn the call over now to my dear friend, Mr. John Molson, and he has an incredible guest. You're going to want to sit back, take notes. I took notes yesterday. I posted some of those notes on Facebook yesterday afternoon. And I have to tell you, I thought I was impressed with what Immunical had done for me and for my family over the years. But after you listen to this gentleman, you're going to get a little bit more of an education than you thought you. If you thought you knew everything about Immunical and were afraid to ask, this would be a time where you're going to know more about Immunical than you ever imagined. So let me turn the call over at this point to my good friend, John Molson, and he will introduce our guest. John, take it away.
Thank you, John. I'd like to begin by thanking all the people on the call today for taking the time to be with us. I'd like to especially thank Keith and John for putting this series of Zoom calls together. It's been a great platform for us to highlight important aspects of Immunotech's unique story. And on the last Zoom call, I closed my portion with the following thought. If you look at the events that led to the discovery of Immunical, the formation of Immunotech, the commercialization of the product, it happened because highly talented people worked together as a team towards a common goal. Now, this is a great lesson because nobody has all the answers. So surround yourself with people who share values, passion, and vision as we did, and great things can happen. Now, ladies and gentlemen, this could not be more on point when discussing the contributions made by our next speaker. On Friday, January 23rd, 2009, at 1:27 p.m., I received the following email from him. Thank you for the interesting information on Immunical. I would definitely be interested in obtaining some of this initially to try some in vitro experiments in primary neural cultures to see what effects on glutathione status and neuroprotection we observe in cell culture models of neural apoptosis and oxidative damage. Please let me know how to go about obtaining Immunical for these types of experiments. So little did I know at the time when I was reading that email the critical role this scientist would play in helping us develop our research platform on Immunocal and brain health. Besides publishing a wide array of papers, he has contributed to 2 of our patent applications, one in traumatic brain injury, and the second one based on our Reelin data, which you'll hear about later. He's also presented Immunocal data at a number of conferences, including the 2018 Alzheimer's Association International Conference, the 2018 Annual Traumatic Brain Injury Conference in Molecular Biology, as well as the Keystone Conference, where he presented our ALS data. Dr. Linseman is a professor of biological sciences and associate executive director of the Nobel Institute for Healthy Aging at the University of Denver. Dr. Linseman teaches courses in neuropharmacology and cell signaling and performs research focused on neurodegenerative disorders and neurotrauma. He received his BS in biology and chemistry from the University of Michigan in 1987. He then worked 8 years as a research technician at the Upjohn Company in Kalamazoo, Michigan. He went back to the University of Michigan in 1996 and completed his PhD in pharmacology. He did his postdoctoral training from 2000 to 2005 at the Denver VA Medical Center and received his first independent VA grant in 2006. He held a joint appointment with the VA and the Denver University from 2006 to 2015 and has been full-time on the faculty at Denver University since 2015. His professional affiliations include the Society for Neuroscience and American Society for Biochemistry and Molecular Biology. Dr. Linseman has published 76 times to date, with many more to come based on his scientific curiosity. Dr. Linseman is a highly valued member of our Scientific Advisory Board. Ladies and gentlemen, it gives me great pleasure to welcome Dr. Dan Linseman to our Zoom call today.
Hi, John. Well, thank you very much for that really kind introduction. And I want to thank John for inviting me today, and thank you all for attending. I've been working for quite a few years on oxidative stress and neuronal apoptosis and how those pathways are involved in neurodegeneration and neurotrauma. And so Immunocal was an obvious fit for this research. So what I'd like to do today is to take you through some of our published works and then also through some data that is not yet published in an Alzheimer's model, so that you can get an idea of the kind of the vast experiences that we've had studying Immunocal. So I'm going to share my computer screen here. And hopefully you can all see that. Yes?
Yes, we can, Doctor.
Great, great. So this is one of the first studies that we started years ago. I can't believe that 11 years has gone by, really. But we initially were looking at in vitro models or cell culture models to see how Immunocal, number one, could it protect nerve cells or neurons from different stresses, particularly oxidative stress? And if so, what was the mechanism underlying that neuroprotection? And so I think with this, I'm going to start actually with the last figure in the paper so that I can give you an idea of what the summary really is. And so my laboratory focuses a lot on the mitochondria and the role of mitochondrial oxidative stress in neuronal cell death, in neurodegenerative disorders, and neurotrauma. Many different types of insults or stressors can impinge on mitochondrial function, which are really your powerhouses of the cell. And this damage that's done to the mitochondria not only decreases energy supply, but it also causes oxidative damage, which can lead to cell death. And what we showed in this paper, sort of in a nutshell, is that Immunocal, by providing a precursor, cysteine, for glutathione synthesis, actually suppressed neurotoxicity or provided neuroprotection from a large array of stressors that act primarily at the level of the mitochondria. So this includes things like free radicals like nitric oxide and peroxide, heavy metal toxicity like that induced by copper, aluminum toxicity, which aluminum is a very well-known neurotoxicant, as well as toxicity caused by, in this case, amyloid beta peptide. Some of you might be familiar with that. This is the peptide that makes up amyloid plaques in Alzheimer's disease. What I want to talk about briefly, just to show you a little bit of the data from this study, is this here. It's called a BCL2 inhibitor. BCL2 is a key pro-survival protein that protects the mitochondria from oxidative stress and really limits what we call neuronal apoptosis, or programmed cell death of neurons. Now, I can show you some pictures of what happens when we treat neuronal cultures with this BCL2 inhibitor. So in the left panel here, these are neurons actually taken from rat brain. These are stained for the nucleus, so this is the DNA, the chromatin of the cell. In the middle panels are the neuronal processes, so these are the networks of connections between these neurons in the dish. And the right-hand panel is the merged image, so the blue are the nuclei and the green are the neuronal processes. And what you can see in this first panel, these are healthy control neurons, and you can see they form this vast interconnected network, and these cells are very healthy. What happens when we add a BCL2 inhibitor, I think it's quite dramatic, right? The cells essentially get destroyed. The nuclei become very small and punctate because they're undergoing apoptosis. And the neuronal processes essentially are completely disassembled, right? So now there are no connections between neurons. But this next finding is really spectacular in my mind. I've been doing these kind of studies for about 20 years, and I've never seen something that protected neurons as well as immunocal. So here we treated the cells with immunocal and the BCL2 inhibitor, and you can see all of the neuronal processes are maintained and the nuclei are still large and healthy. And so as we would predict, what happens if we measure glutathione in these neurons, we see that in response to the BCL2 inhibitor, we get a dramatic reduction in cellular. Okay, we're back. Okay, I apologize for that. I don't know what happened there. So what I was saying here is that we were trying to look at the mechanism of how immunocal protected these neurons so well from this BCL2 inhibitor. And what we found was that, as predicted, this compound dramatically depletes cellular glutathione and induces tremendous amounts of cell death. However, when we treat with immunocal, we preserve or protect that cellular glutathione, and that, of course, then reduces the cell death dramatically and protects the neurons. But how do we mechanistically show that those two events, the preservation of glutathione and the neuroprotection, are connected, right, mechanistically? Well, in a cell culture system, you can do that because we can put in a compound here, BSO or buthione sulfoxamine, that actually inhibits the synthesis of glutathione from cysteine. And when we put in that compound, you can see it blocks the ability of immunocal to preserve glutathione, and you lose the neuroprotective effect completely. And so this was really the first study in vitro that we were able to very convincingly show that the ability of immunocal to protect neurons from different types of oxidative stress-inducing agents is absolutely dependent on its ability to stimulate or preserve glutathione synthesis. So we went on from these in vitro studies to do a number of different studies in mouse models of various either neurodegenerative disorders or neurotrauma effects. So the first study that we did was in a mouse model of amyotrophic lateral sclerosis, or ALS. This is a transgenic mouse that expresses a human mutant transgene of the SOD1 gene. This mutation is found in a small percentage of familial ALS patients. So what we did is we gave these mice immunocal either as a direct, what we call oral gavage, where we basically put a tube in the mouse's mouth and deposit the immunocal directly into the stomach, or we gave the immunocal in their water so that they could just drink it on a daily basis. And either way, we got the same effect in this mouse model. What we saw was about a 10 to 12 day delay in the average onset of the disease in these animals. And we measure this onset through changes in motor function, as ALS is really a neuromuscular disease that causes deficiencies in motor function. Now, a 10 to 12 day delay in disease onset may not sound very significant, but you have to understand that in a mouse, 10 to 12 days is really roughly equivalent to 18 to 24 months in a human. So this is actually a very large effect in this mouse model. We also looked at motor function in these animals, and one of the ways that we test motor function is by something called a grip strength test. Basically, you take the mouse, you put it on a wire grid, and you invert the grid and see how long the animal can hold on with its hind legs, its hind paws. Now, mice are tremendously adept at this. This top line are the wild type or non-transgenic healthy mice. We stopped this assay at 30 seconds, but they could hold on for hours, literally. They climb around on it upside down. They have no issues. This line here, though, are the mutant transgenic mice that have ALS that were untreated. And you can see by the time they're about 105 days old, essentially these animals have no strength in their hind limbs, and they're not able to hold on to the wire grid. And this is really due to a loss of innervation of the motor neurons into the skeletal muscle in the legs. This is part of the pathology of ALS. What you can see is that the animals that we gave immunocal to, they showed a really nice preservation of motor function here. So this is skeletal muscle strength in their hind limbs. And this is a very big difference. You can see that they sustained really this increased muscle function out to nearly 120 days old, which is normally about the end stage where these animals start to succumb to the disease. We also looked at glutathione in the spinal cord. So this is actually looking in the lumbar spinal cord, which is your sort of your lower back, where the motor neurons project their axons over 1 meter in length to innervate your large leg muscles. And what we were looking at here is we used a technique called HPLC, or high performance liquid chromatography, to measure both the reduced and oxidized forms of glutathione. What we found was that in the spinal cord of these animals, compared to the healthy non-transgenic mice, ALS mice show a marked reduction in the reduced form of glutathione and a marked increase in the oxidized form. This leads to a decrease in the ratio of reduced to oxidized glutathione, and this decrease is really reflective of tremendous oxidative stress in the spinal cord of these mice. But what's quite remarkable is the animals that we treated with immunocal, you can see they showed a preservation of the reduced glutathione and essentially no increase in the oxidized glutathione. And so this led to really a clear protection of the reduced to oxidized glutathione ratio in the spinal cord. indicating that immunocal treatment actually suppressed oxidative stress in the lumbar spinal cord of these ALS animals. So this was a pretty fascinating finding. The last thing that I want to show you from this paper was kind of an interesting study that we did. So if any of you are familiar with ALS, you'll know that it's really a severe neuromuscular disease. There are very few drugs, only 2 now approved for ALS, and they really don't have very large effects in terms of therapeutic effects. The one drug that was approved all the way back in 1995 is riluzole. And riluzole has what we call an anti-glutamatergic action. It basically decreases calcium-mediated toxicity in the motor neurons. So quite a few patients that have ALS, probably about 50% of all ALS patients, are on riluzole. And so one of the things that we wanted to make sure was that if we gave these patients immunocal, that we would not in any way interfere with the actions of riluzole. Even though they're not tremendously effective, there are some positive effects of riluzole. And so what we did is we either treated our ALS mice with riluzole alone or with immunocal and riluzole. And what we found here is that the combination of the two, we still see this delay in disease onset. You can actually see that you do not see this with riluzole. It doesn't have this effect. So this is actually the immunocal effect, delaying disease onset. But more importantly, riluzole does extend survival of these animals, and the addition of immunocal had no adverse effect on the survival promoting action of riluzole, indicating that patients that are on riluzole and receiving some benefit from this drug would not have an adverse effect from taking immunocal on top of that, and that's a very important finding. So that was our ALS studies, and through conversations with John Molson and that, we decided to focus in on neurotrauma. As you are probably aware, concussion and traumatic brain injury is a tremendous unmet medical need in the health community. There's approximately 1.7 million concussions every year in the United States, and there are no drugs approved for the treatment of concussion. Now, we were particularly interested in high-risk populations that are in either occupations or that have a high risk of concussion. So this would be athletes in contact sports, military personnel, as well as the elderly, because the elderly, unfortunately, do have a lot of concussions due to their propensity to fall. And so we actually did a study using a mouse model of concussion or traumatic brain injury, in which we use a model called controlled cortical impact. And basically what this model is that we, under anesthetic, we do a traumatic brain injury to the skull of the animal that would be comparable to somebody having a moderate concussion. So this might be an athlete playing football and having a moderate concussion, akin to maybe just a slight loss of consciousness, maybe for a couple seconds. And what we did is, because we were interested in seeing if immunocal could increase resilience against the negative effects of concussion, we actually pre-treated these mice with immunocal in their drinking water for about 4 weeks prior to giving them a concussion. And then we looked at a whole host of behavioral assays, histopathology, as well as biochemistry to analyze how immunocal affected the response of these animals to concussion. And so I'm not going to take you through all this data. There's a lot of data in this paper, but I do want to highlight a few things. The first thing here, you're going to see two graphs. One is called writing reflex time and the other one is apnea time. So apnea refers to the inability of the mouse to breathe. So once under anesthesia, when we do the concussion on these animals, for about 30 seconds, they stop breathing. Okay, and this is common in these types of paradigms that we use in mice. Writing reflex time refers to how long the animal takes to wake up from anesthetic after the concussion. The sham animals here are the controls. Essentially, they have all the same surgical interventions that we do to the animals that get the traumatic brain injury, except they don't get hit, right? So they are a control, but they don't ever suffer a concussion. What you can see is that the sham animals take a couple minutes to wake up from anesthetic, and of course they don't have any apnea at all. The animals that were untreated that received concussion, you can see they take quite a bit longer, more like 6 to 7 minutes to wake up from anesthetic after receiving the concussion. And they undergo about 30 seconds of apnea. The important thing here is that the animals that were treated for 30 days prior to the concussion procedure with immunocal, they don't have any difference in either their writing reflex time or apnea time from the animals that did not receive immunocal but received concussion. So why would this be important? This is important to demonstrate that both the untreated and treated mice had the same magnitude of concussion, okay, induced. There was no difference. We didn't hit the immunocal animals with a lighter magnitude of concussion, for example. So this is important sort of control data to make sure that it's a fair test of the effects of immunocal. So then we did several behavioral assays. One of those was something that we call a beam walk. And basically this is the ability of mice to walk on an increasingly narrower and narrower beam. Okay, mice are tremendously agile creatures, and they can walk on beams that are very, very thin. We're talking somewhere around 1/8 of an inch. Okay, and so what we're looking at here is the ability of these animals to traverse this beam and how quickly they can do so. What you can see is that the TBI animals have a severe deficit in traversing this narrow beam. And the animals that received immunocal, they perform much better, almost as well as the sham animals in this really challenging motor task. So we saw a benefit there of immunocal on preserving motor function. We also did another kind of similar assay. This is called a rotarod. And basically what this is a wheel. Okay, and so mice love to run on wheels. This happens to be a wheel that goes extremely fast. It's basically a little conveyor belt wheel that we program to go very fast. And so sham animals can stay on it for about 20 seconds before falling off. The untreated TBI mice could only stay on about half as long, but the animals treated with immunocal showed a really essentially the same as the sham mice in terms of their performance on the rotarod. So again, immunocal showed a nice protection or preservation of motor function in these animals that received a concussion. We also tested cognitive function in the mice. Now, it can be challenging to look at the cognitive ability of a mouse. We typically do this with mazes. In this case, we use what's called a Barnes maze. You can think of a Barnes maze as a circular platform with about 18 fake holes on it. However, one of those holes actually is real and has a little compartment that the mouse can escape into. It's called an escape pod that is below the platform. So what we do is we put the mouse in the middle of this platform and we shine a bright light on them. Mice don't like to have light shined on them. And then the mouse will try to find that escape hole. And we train them in what's called an acquisition phase over a number of days using spatial cues, basically an arrow that is drawn on the wall pointing to the escape pod. And over time, these mice learn where the escape pod is. What you can see, though, after 6 days of this training paradigm, the sham animals take about 40 seconds to find the escape pod, but the animals that received a concussion take nearly twice as long. Okay, so this is actually a measure of what we call spatial learning and memory. The animals that receive the concussion do not learn as well. But what's interesting is animals that were treated with immunocal and then subjected to concussion, they learned just about as well as the sham mice. And so this was a nice protection, if you will, of cognitive function. We also did what's called a probe phase. This is a sort of an acute, after a week of training, we just put the mice in there and we do a certain test to see how quickly they can go to the escape area, but not necessarily enter the escape pod. And you can see that the TBI animals, the ones that had concussions, they take about twice as long as the sham mice to find that escape pod area, whereas the immunocal treated animals, they actually were a little bit better than the sham mice at finding the escape pod. So immunocal pre-treatment prior to concussion actually preserved cognitive function and prevented these cognitive deficiencies in the mice due to concussion. We also looked at glutathione in the cortex. So this is in the cerebral cortex, the brain of these animals. And similar to what we found in the spinal cord in ALS mice, we see with concussion a reduction in the reduced glutathione to oxidized glutathione ratio, and that reduction is prevented by immunocal administration. So that's consistent with the mechanism that we see in vitro for neuroprotection as well as what we saw in ALS. We also looked in the brain at an index of oxidative stress. This MDA is a lipid peroxidation product that is markedly increased in the brain of animals that receive a concussion, and the levels of this lipid hydroperoxide are significantly decreased by immunocal treatment, indicating that the immunocal is protecting the brain from oxidative stress. Another important thing that we looked at were levels of a certain neurotrophic factor. This is a growth factor for nerve cells. It's called brain-derived neurotrophic factor, or BDNF. And it's an important factor because it protects neurons from different types of damage. And it's considered a pro-survival factor. And what we found was that mice subjected to concussion showed a marked reduction in both the pro or inactive as well as the active forms of BDNF, and that this was protected or prevented, this reduction was prevented by immunocal treatment. So immunocal not only reduced oxidative stress, it also protected this key pro-survival growth factor in the brains of these mice that had concussions. We also looked at several indices of what's called neuroinflammation. So in this case, inflammatory cells in the brain like microglia and astrocytes, once there's an injury, they enter that site of injury and they can cause additional oxidative stress and inflammatory stress. So in this top panel, what you're looking at here is the expression of a protein known as IBA1, which is a marker of microglia, which are an inflammatory cell in the brain. what you can see is that animals that were subjected to concussion have much higher levels of IBA1, so they have much higher microglia, and this level of IBA1 is diminished dramatically by immunocal. And so this would argue that immunocal actually has an anti-neuroinflammatory property in addition to having antioxidant and neuroprotective effects. I just want to make sure you guys can hear me still.
We can hear you, doctor.
Good, good, okay.
Taking notes.
Okay, good. Okay, the last two slides I want to show you, these are more looking at histology and looking at the damage in the brain that occurs in response to a concussion. This first image I'm going to show you, this is what's called the corpus callosum, which is a region in the brain where many of the white matter axonal tracts of the neurons cross. So the brain is made up of gray matter, which are the cell bodies of the neurons, and the processes, particularly the axons, which are myelinated, make up the white matter or the white matter tracts. And these axons, myelinated axons, are very important because that's how nerve impulses are conducted from cell to cell in the brain. We can stain these white matter tracts with something called Luxol Fast Blue. And so in a healthy sham mouse, you can see a nice large distribution of these white matter tracts. Interestingly, in animals that receive this moderate concussion, several days, this is about 2 weeks after concussion, you can see many of the mice lose a fair amount of these white matter tracts. And this is due to degeneration of the axons or the processes of the neurons, which of course will cut down on conduction of nerve impulses in the brain. What you can see with immunocal is that largely these reductions in white matter tracts were prevented by immunocal administration, and that's shown quantitatively here. So immunocal pretreatment actually protected the neuronal axons or processes from damage. The last slide I want to show you is we looked directly at degenerating neurons or nerve cells in the brain of these mice beneath the impact site of the concussion. And we did that using a dye called fluorojade, this green dye, that only stains neurons that are degenerating or dying. And what we found was that in the brains of mice subjected to concussion, we would see foci, these areas, discrete areas where neurons were degenerating. And so we compared the fluorojade positive foci in untreated TBI animals to animals that were treated with immunocal. And you can see that immunocal really significantly protected these neurons from degeneration in response to a concussion. And so this series of studies really demonstrated that pretreatment with immunocal has a really nice protective effect, increasing the resilience of the brain against a moderate traumatic brain injury. And I could mention that we're in the process of completing a study right now where we're doing a similar type of scenario, except we're using a model of repetitive mild traumatic brain injury. So this is more like somebody who maybe gets a series of concussions, maybe an athlete that over the course of a couple seasons gets 3 or 4 concussions. So it's a little bit of a different model, but I can tell you we're seeing significant benefits of the immunocal administration in that model as well. The last study that I want to talk about is actually not published yet. We're in the process of writing this up and submitting it for publication. And this is looking in a mouse model of Alzheimer's disease. And so this again is a transgenic mouse that has expresses a human mutant gene. It's called the amyloid precursor protein gene. and this is the Swedish Indiana mutation of this gene. The amyloid precursor protein is responsible for generating the amyloid beta peptide that ultimately forms amyloid plaques or senile plaques in Alzheimer's disease. And this particular form, the Swedish Indiana mutation, leads to a high turnover rate of this amyloid precursor protein into this toxic amyloid beta peptide. So these mice, they actually develop amyloid plaque pathology just like humans that develop Alzheimer's disease. And typically it takes somewhere between 8 to 12 months for these animals to develop this Alzheimer's-like pathology. So what we did is we began giving these animals, these mice, immunocale in their drinking water when they were 3 months old. And we gave that to them until they were 12 months old. And at that point, we analyzed their brains for biochemical, histological changes that are similar to what you would see in Alzheimer's disease in a person. And so let me start with the measure of glutathione in the brain of these animals, because that's really what we're talking about mechanistically here. And so I can just show you the ratio again. This is the ratio of reduced glutathione to oxidized glutathione in the cerebral cortex of these mice. So the non-carrier mice, these are non-transgenic control mice. These are healthy mice. These hemi-untreated mice, these are the transgenic Alzheimer's mice, untreated. And then these are the transgenic Alzheimer's mice that were treated with immunocale. And similar to what we saw in the spinal cord in ALS or in the brain following TBI, in these Alzheimer's mice, you see a dramatic reduction in the reduced to oxidized glutathione ratio in the brains of Alzheimer's mice. But immunocale treatment over that 9-month period of time essentially prevented that and dramatically preserved the glutathione in the brains of these animals. Now, John had mentioned a patent, something that we were looking at with this protein reelin. Reelin, let me explain what that is. It's a protein that's involved in development of the brain. However, once the brain develops, it plays a key role, particularly in a region of the brain known as the entorhinal cortex and the hippocampus, in inducing something called long-term potentiation and synaptic plasticity. These are processes that underlie learning and memory. And so reelin is released by cells in the entorhinal cortex into the hippocampus, where it signals these cells to facilitate learning and memory. Interestingly, one of the first things that happens in Alzheimer's disease is that reelin expression in the entorhinal cortex goes way down, it decreases. And this likely contributes to the loss of cognitive function, the decreased memory formation that we see in Alzheimer's patients. And so if we could preserve reelin expression, that might go a long ways towards a new therapeutic approach for something like dementia. And so in this first panel here, what I want to show you is we're actually doing a process that's called in situ mRNA hybridization. Here we're actually looking at expression of the messenger RNA transcript for reelin, which will tell us which cells are actually making the reelin protein. Later on, I'll show you staining for the reelin protein, but there reelin is kind of everywhere because it's actually a secreted protein, and so it's all over the place. But in these, it's much more discrete expression because these are the cells that are actually making the reelin. What you can see in the healthy mice, so the blue here are the nuclei again, or the chromatin, just showing you all the cells in this region. And the green is stained for the reelin transcript. what you can see is that in the healthy mice, in the entorhinal cortex, there are a lot of cells that express the reelin transcript. However, in the 12-month-old Alzheimer's mice, you can see that essentially the reelin has been depleted from this region of the brain, which is what we see in human Alzheimer's patients. However, those animals that were treated with immunocal show some preservation of reelin expression in the entorhinal cortex, and that's shown here in this graph. And so immunocal is able to actually protect reelin expression in the entorhinal cortex. We also looked at this by a technique called Western blotting, where we're looking at the actual reelin protein. So that's in this top panel. So you can see reelin is expressed in the hippocampus entorhinal cortex of the control mice. It goes down in the Alzheimer's mice, and immunocal, again, protects the expression of that reelin. We also stained the brains of these mice to look at reelin protein expression. And so here the green stain is reelin. The left panels are the control mice, the middle panels are the Alzheimer's untreated mice, and the right panels are the Alzheimer's mice treated with immunocal. And here we're looking at entorhinal cortex, and here dentate gyrus, which is the region of the hippocampal structure that the entorhinal cortex reelin-expressing neurons deposit their reelin into. And what you can see is that the Alzheimer's mice show marked reductions in reelin expression in comparison to the control mice, but immunocal treatment really preserves this reelin expression. So you can see that here quantitatively, that these reductions in reelin are actually prevented, largely prevented by immunocal treatment. So not only does immunocal preserve glutathione in the brains of these Alzheimer's mice, but it also preserves reelin expression. The last thing I want to show you, because these animals have this mutation in the amyloid precursor protein and make a tremendous amount of the amyloid beta peptide, they actually form amyloid plaques. And we can look at those amyloid plaques using a compound known as amyloglow that sticks to the plaques and makes them light up. And so this green staining in the different regions of the hippocampus, the dentate gyrus, as well as the entorhinal cortex, these are amyloid plaques that have formed in the brains of these 12-month-old Alzheimer's mice. And you can see in the untreated mice, there are a large number of these plaques. In comparison, here's an immunocal treated mouse. There still are a few plaques, but they are much smaller and take up much less area in the regions of the hippocampus. And we quantified this both by the plaque number, so the absolute number of amyloid plaques, as well as the plaque load, which is basically the percentage of the area of the hippocampus that is taken up by amyloid plaques. And what you can see essentially in all cases is that immunocal treatment resulted in somewhere between a 50 and 80% reduction in the amyloid plaque load in these animals, which is really a tremendous effect in this model of Alzheimer's disease. So the last thing that I want to mention is that in addition to these studies that we have published and that we're working on publishing now, we've also published a number of review articles and editorials. This one was on using immunocal to preserve glutathione as a novel neuroprotective strategy for the degenerative disorders of the nervous system. We also published this editorial or perspective article on glutathione precursors like immunocal shielding the brain from neurotrauma. And we also had this article that we published on detailing really the effects of immunocal in this mouse model of traumatic brain injury. And I think really to kind of summarize what we've shown is that immunocal, through its ability to provide a precursor for glutathione synthesis, its effects in the brain and spinal cord are to increase resilience of these central nervous system areas to stress and disease mechanisms. So by preserving that glutathione, we can reduce oxidative damage, we can diminish neuroinflammation, we can preserve neurotrophic factors like brain-derived neurotrophic factor. We can also preserve key cognitive proteins like reelin. And so this combination of effects that immunocal has really leads to really a nice potential therapeutic option, as a nutritional supplement for people. So I think I'll stop there, and if anybody has any questions, be happy to
one
answer.
question, Dan, for everybody. First of all, it's fantastic. I just want to tell everybody this has been 10 years worth of work, and Dr. Linseman is the ideal partner because he's completely reliable and completely trustworthy, and every time I phoned him, he was always there to provide us the information, and it was always dependable. But then just in the context of these terms we use, if we talk about resiliency and we talk about unhealthy brain aging and healthy brain aging, how would you talk about the term resiliency in terms of unhealthy brain aging versus healthy brain aging, in discussion with immunocal and what you've seen immunocal do?
Yeah, so I think that gets right at the heart of one of the main theories for aging, and certainly brain aging, is it's known as the oxidative stress theory of aging, where as we age, everyone has some, decrease in our endogenous antioxidant defenses. That happens over time as we age. In addition, the mitochondria, which I mentioned are the energy producing systems in cells, they're somewhat inherently inefficient, and one of the products of mitochondrial function is a little bit of a leakage of electrons to oxygen to create free radicals. When we're young, our mitochondria are much more efficient, and we have good antioxidant stores, and so we can really limit that free radical damage. But as we get older, our mitochondria become much less efficient. They leak more electrons. We get a higher free radical load. And in addition, we have a depletion of our antioxidant reserves. And so you've got kind of two things going on there. You've got increased oxidative burden, but decreased antioxidant reserve. And this, of course, is most pronounced in the brain, because the brain, because of the way that neurons communicate and their dependence on high energy levels for neuronal communication, the brain is tremendously dependent on energy stores and is also ultra sensitive to oxidative damage. And so I think having a supplement that boosts that endogenous antioxidant pool by, in this case, providing a glutathione precursor, because glutathione pools are actually depleted as we age. That's a key way to counteract this sort of aging effect. Now, you mentioned unhealthy aging. I mean, that's just normal aging, but unhealthy aging comes in the context of other diseases that underlie the aging process. So that can be due to your particular genetic makeup, can also be due to environmental stressors. We mentioned yesterday emotional stressors, but also trauma. So if you have a history of concussion, that can lead to enhanced oxidative damage. And of course, the oxidative damage feeds into neuroinflammation, because oxidative stress triggers increased inflammatory responses in the brain, process we call neuroinflammation. And then that feeds forward in a cycle because neuroinflammatory cells produce free radicals as part of the mechanism of inflammation. And so you have sort of a vicious cycle there. So I think, having on board a supplement that can preserve and stabilize your antioxidant pools would be key for healthy brain aging.
Great, and I'll close it there and just say thank you on behalf of all of us. It was great to listen to, and I'll hand it back to John.
Okay, just a second. I don't see John here. We'll get him. There he is. All right. John, you're still muted.
Okay,
There you go.
All right. Dr. Linseman, amazing stuff. We're going to make a little change in the batting order here. Our CEO is with us, and unfortunately, he's only got a few minutes. He's got another appointment he has to get to. And I just have one real fast question as I introduce him, and that is, Mauricio, if you can just kind of tell all these wonderful people around the world that are listening to you today, when you purchased the company some 3.5 years ago, how much was your decision based on research by people like Dr. Linseman, of course, and all the other great researchers that have worked on this Immunical project for some 40 plus years? So I'll turn the call to you and let you answer that for everyone. And welcome to the call, Mauricio.
Thank you, John. Thank you as always. And thank you everyone for joining us into this amazing Zoomtacular. I think it's now probably the 10 or 12 that we have been doing together. And isn't it amazing what we have heard from Dr. Linseman? I really, really enjoy it. I think with you all. But the most important thing about of these conversations is that you can tell how that we're committed to our scientific heritage. It is very important for you to know, obviously, for those that are doctors, I think that you enjoy it even further because you understand way better than what most of us didn't. And but the most important thing for you to know is that we as Immunotech are doing the heavy lifting on that end. We're gonna keep committed, and we're gonna keep doing every single thing that needs to be done into the science and into making sure that you are showing and you are sharing this amazing product with whoever you want. And you don't need to be a doctor. If you are, welcome to our family. If you're not a doctor, you don't need to be a doctor. You really need to just know that we're going to be working hard into our scientific heritage. And that's our DNA. That's part of our DNA. And it always be. We will never get away from that. We are committed. This is the only multi-level marketing company in the world, I can tell, that invest and do that many research without being able to share with the people that many research. Because we can't, unfortunately, because the way we are, the way our product is commercialized, we cannot share that many information. But incredible people that like Dr. Linseman, obviously like John Molson that has invested his whole life in this amazing project as well, like Dr. Gutman and many, many, many more, obviously Dr. Conchavan and Dr. Bunos since the beginning, but many, many more, because you need to know there's many doctors involved. We even have our first scientific meeting. Probably John Molson is there. I cannot recall exactly the day, but probably 2.5 years ago, we had our first summit with all our scientific board, because by the way, you all, if you don't know, we have a scientific advisory board that is part of our company, and that's just for you to know that we're committed to that. And that's what we believe in John Molson since day one that we starting these conversations. One of the things that excites me the most is how deep into the science we were. But the better thing on that is that we were doing the heavy lifting as a company, and you as part of this amazing family, you just need to share this product because we are doing that part. We have people as committed like John Molson, like as I mentioned, several others that are committed to this product, are committed to the scientific research, and that will help us to be more secure, to be more sure that we can share this amazing opportunity with this amazing product that helps thousands and millions of lives. As I told you last Zoomtacular, at this time, at this point, every single second that passed, we have sold 3 pouches of Immunocal every single second of the 24 hours. So imagine how important it is for us and how committed is our people to the scientific portion. So we will continue to do that. And obviously, John Solder, that took a lot in our decision, in my decision, in the acquisition of this company, because as we have talked many times in the past, and I will not mention any company or any other because that's the way it was, but in the past, many people were saying that it doesn't matter which product you're selling. Even if you have a good compensation plan, you don't need to. You don't need to have a great product. You don't care about the product. Well, guess what? In Immunotech, we do care about our products. We do care about our products. And that's why we have invested over $25 million, over 40 years of research and development. So that is how committed we are. Imagine having people like Dr. Linseman in this call for that long, that he has done amazing discoveries and amazing research for many other things. And it's part of our scientific advisory board. It's one of the great members that we have. So I will not go into the science because that's why we have those experts doing that. But it's amazing, and you need to know that we are gonna be committed, and we continue committed on that end. John? I think John Solider is in.
He may have muted himself again, Maurizio. So let me get back here and see if I can.
Nice to say hi to you, Kev.
Well, glad to have you here. Let me ask John to. He keeps.
It's not me. It's the old guy running the call.
Oh, listen. See Maurizio. See what I have to put up with.
He knows. He knows. There's not a lot of technology between the two of us. Anyway, thank you so much, Mauricio. I know you got another engagement to run to there, so always appreciate that. But you know, it just shows once again, just to reiterate before Mauricio goes, and Dr. Linseman, amazing. I've got 8 pages of notes, and I had 4 yesterday. So if I listen again tomorrow, I'll have 16. I'm actually learning this stuff. But, you know, it just goes back, just to reiterate, you know, obviously Mauricio and Dr. Linseman are fully aware of this. But for those of you who are listening for the first time, 1975 collaboration between 2 McGill University researchers, Dr. Patricia Kongshaven, Dr. Gustavo Bunos. 1978, the first paper written on Immunocal. You know, contextually, you know, think where you were in 1978. Some of you weren't born. 1980, some money gets invested in the company.
That's the age I born, my friend.
I was only a freshman in high school in 75, so it makes me feel a little better.
I'm actually 78, exactly like the same day this company was published, the first one. Yeah.
Isn't that amazing? Amazing. And then you think about it, 1996. But what happened in that time frame between 1975, 78, and 96 is significant also, because, you know, Dr. Linseman is too young to have been involved at that point, but we had researchers like Dr. Luc Montagnier at the Pasteur Institute, Dr. Wolf Droge working up in Heidelberg, Germany, people from Otsuka Pharmaceutical over in Japan, et cetera, et cetera, et cetera. By the way, Dr. Montagnier was the co-founder of the HIV virus, won the Nobel Prize, to give you an idea of the stature of some of the researchers that we've had, like Dr. Linseman and others that you're going to meet in coming Zoomtaculars, working with Immunocal. So there's never been, when Mauricio says, and when I say and others say, there's never been a network marketing company like Immunotech based on evidence-based science 21 years before anybody said, hey, how do we make some money to market? Just think about that for a minute. 2 decades without anybody earning a nickel from marketing this product. That's the commitment to the science. And that commitment, more importantly, since Mauricio purchased the company, has continued and will continue going forward. You heard it from him today. So what we're going to do at this point, bring another one of our medical friends on. who happens to also be an immunotech distributor way over in the United Kingdom. And he's going to give us some idea now how to monetize some of the information that Dr. Linseman shared this afternoon. In other words, how to take this out to the marketplace and communicate it, whether you're talking to a truck driver, a school teacher, someone in a restaurant, or for that matter, your chiropractor, your MD, your dentist, your Reiki specialist, your naturopath, whomever, so that you can take this information and actually communicate it in such a manner that somebody says, okay, get me that product, let me start using it. So let me turn the call over now, and I hope he's there, I believe he is, to my good friend, Dr. Jeffrey McTavish. And Dr. Jeffrey, if you can shed some light on how to take this product now to market in an effective way.
Thank you, John, and hello, everyone. Dr. Linseman, that was a very, very informative information that you gave us, and it really cements a lot of things that I have gone through in my training as a chiropractor and doing also some postgraduate work with functional neurology. And it sort of cements the whole idea about, and I'm just going to use some chiropractic sort of terms and analogies, is that when we explain to people that you've got inflammation, you have maybe have some inflammation in the spine that can create pain, and people come in, they've got back pain, they've got neck pain, or they may have pain somewhere else, is that the ability of the Biochemical product to address these particular items. When we look at presenting this product to my colleagues or talking to a dentist or a medical doctor, one of the greatest things that we've got with this product is that it has such respect in the scientific world for its ability to deliver consistently a product that will deliver an answer of making glutathione. And that's the really cool thing about this thing. We've had loads of studies over the years with HIV, AIDS, cancer, cystic fibrosis. It's been tested with some sports performance papers. It's so respected in the scientific community that we look at it with revere and respect, because let's face it, it has a, maybe we have to remind ourselves sometimes that it has that natural health product number in Canada, which is, from what I understand from speaking to John Molson and some other people, very difficult to get, because that then is telling us that the product is safe, it's effective in its design and what it's supposed to do, and it is at the highest quality. And this is why it probably gets the respect that Dr. Linseman is using, because of the fact that it will deliver the results that he's trying to hypothesize in his particular studies. And that is the type of research and quality of the product to take to my colleagues and say, this is what this product can do. And we have such a wonderful backlog of information that is scientifically validated that we really don't have to say we're talking out of our butt, because this actually works and is proper. And the other thing what I would say is that when we have all of this strong scientific data, sometimes for some people it's very difficult to talk to Mrs. Jones or Bob down at the pub. Then the idea being is that because we have the data to show that it works, we could just say it works. You don't have to understand the science unless you're a geek like me or somebody who wants to know it. And then we can simplify it very easily. So to give you sort of a context, when I'm dealing with somebody who's coming in and they've got some problems with the spine, I can look at various motoric output tests, normally using the eye, to find out whether or not there's going to be some sort of problem within the brain on a physiological level. And this is what we would call transneural degeneration. And we'd be looking to see the changes that happen within eye movement or any sort of cognitive change that would constitute a variation of what should be normal. And we can then hypothesize that possibly they've got some transneural degeneration going in the brain that will give us this particular output. And what's really cool is that Dr. Linseman just nailed it for me with this presentation. It's there in his studies showing me what this functional neurology people have been talking about for years, is this whole thing about how the brain is affected by oxidative stress over long periods of time. And this is the thing that I think we could take home to anybody really, is that term inflammation or oxidative stress, or what we're talking about is the mitochondria, because you have to think about, if you've got one brain cell, you may have up to 3,500 mitochondria, all producing this hydroxy radical or these free radicals. And our ability to counteract that over age, particularly when we're doing it in decades, because then we have to look at your particular autophagy rates and your haplotypes, and there's so many different variables that we could look at to then constitute whether or not, if you're 61 years of age like myself, am I physiologically younger or am I older? And we can then use some tests to find this out. So we can look at how the ability of Immunical to protect the very thing that keeps us alive, and that's this here. And that's the stuff that runs down your back. And it's called the spinal cord. So when we look at from a point of view from chiropractic, we would look at something like this. I don't know if you can see this, but this is a real, there, can you see that there? It's a real bone. And the idea being that when our joints go out of alignment, it can create a lot of inflammation in the joint, which then creates you pain. So the idea, what Dr. Linseman had said, was the fact that they found changes within the neuronal pools within the spinal cord. Now this is really important for us as clinicians. And then you could take it one step forward and bring in some people, say like the dentist, because there's some really good data out there to show that mercury can actually cause neuronal death quite quick. So all these people who've got the amalgam fillings in their mouth, we can measure the amount of mercury that's in the body, which can then be purportedly then looked at how much inflammatory markers they have in the body as well. But we could also look at their glutathione pools, and looking at things like you can do whole blood, so you can do glutathione peroxidase and reductase. So there's different elements that you can do in blood tests to find out how much Immunical these particular people would require in order to bring up their glutathione pools within not only the brain, which was today's subject, but also in all the other tissues in their body that will require glutathione. The whole thing is taking this to people to say what we could do to them to see how this could be so good and neuroprotective. It's very, very simple. Is you could probably take them to some sort of a home where they deal with dementia patients or Alzheimer's patients, and it becomes critically really clear. I've got one next door to me right here, and they have dementia patients in there. All I have to do is open my window and listen to some of them go screaming because they've lost it. And this is the thing that we're all trying to avoid, is that this thing here that actually keeps us alive and going. We want to make sure that that prefrontal cortex can still compute all of our data and make rational decisions, so that we don't go wandering down the seat butt naked and doing this, going, nah, 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 because this is what we don't want. And so to have a look at the fact of how Immunical can help, what we want to do is be able to stress the fact that it is neuroprotective. And it does that by augmenting the glutathione within our cells. And if we then think, okay, the engines of our cell are called the mitochondria, and those things are abundant in the brain and in the heart as well, but they're everywhere in our cells. So a muscle cell may have 500 of these, and they're all producing free radicals. And it's the ability to downregulate free radical pooling and have a negative effect on the nervous system and any other part of the cell that may be affected by those. So when it comes to, John, to answer your question about taking this to people and talking to them about how Immunical can help, the biggest thing is that it's neuroprotective. It's protecting our brain. It's protecting our spinal cord, which is giving us the information from the brain to the body. With that, it also is going to protect all the organs that need neuro information to come up to the brain, so the brain can modulate how that organ, that liver is working, how that gallbladder is working. All the other bits and bobs in our body need to communicate to our brain. But if our brain health isn't as up to speed because we have problems processing information, or we've got problems, again, from a chiropractic level, problems at a spinal cord level because of the vertebrae causing inflammation, then we're going to have a change in how that information gets exchanged between the brain and the body. So by reducing inflammation, and there's a key word in the word inflammation, and that's flame. A lot of people, when they have a sore back, they put heat on it. And actually what you want to do is you want to put cold on it, because you want to dampen down that inflammation, dampen down the free radical pooling that's happening within that region. So by just showing basic things like that, I tend to use, for me, thermal imaging is very easy to show people by taking a picture of a back or a face or a hand and indicating neural stress within a joint or in a back and inflammation as well. And one of the things that we can then tell people is that if you use a product that's natural, that has such scientific clout, but also consistency. So every time you take it, you're going to be taking a product that can give you and deliver all the time the precursors to making glutathione all the time. Then we can say to people with our hand in our heart, this product will raise your glutathione levels. Glutathione is neuroprotective. It's going to help your brain. It's going to keep you intact neurologically with all the insults that we're all exposed to, like Wi-Fi, mobile phone technology, and the LED lights that are coming out that have a detrimental effect on our mitochondrial function. So there are many, many reasons why we need to have this particular product. Everybody has a different environment they're exposed to. So sometimes the need is greater for some people than other people. But we all need it. We have to have this in order to make a robust immune system, to help detox our systems, but also have this ability to have neuroprotective effects so we don't go doolally later on because of the degradation of our central brain nervous systems and lose our ability to recall memories. That's the way I would bring it across to people, and stress the fact that the more you take, the better protection you get. So a lot of people say, well, how much should I take? And I say, well, if you want, you can get your bloods done and find out, or we can muscle test you, or you just start taking it and then see what type of changes happen with the problems that you particularly may have or complaints that you have, et cetera, et cetera. Just get on the product because we know it's going to do its job. And we know that because of the consistent data from scientific studies all the way up to just Mr. Jones taking it and feel good when he wakes up the next morning, or he's going to run a marathon, or whatever it is the situation is. We know that this product will work for us, given time, every time. I think that's Keith Hooper's good acronym word there. And John, that's how I would say it to people. I mean, it really depends on who you're talking to. So then you steer that conversation according to who you're talking to and what kind of data you want to give them.
You know, as I listened to you, Dr. Jeffrey, and I listened to, Dr. Linseman and Dr. Kongshaven a few weeks ago, what occurs to me as a layman is the hard work, the heavy lifting. I think Mauricio's word a few minutes ago, the heavy lifting has been done by the scientific community and validation, starting with Dr. Kongshaven, Dr. Bunos all those years ago, 1975, when most of us were either not thought of or very young. And of course, continuing today with Dr. Linseman and several other researchers that we're going to mention here before this call is done. So do not get off. You do not want to get off this call right now, folks. We've got about 20 minutes to go and a lot more information that we're going to share with you. But, once again, for me as a layman, my expertise is in marketing. So I don't want to have to understand all that science that you understand. And obviously Dr. Linseman understands, at such an incredibly high level, and the other researchers do. All I want to do is I want to get up in the morning and take something that I know does all of that, and that somebody else really, really smart in that category has done their job. And that's what we have here with Immunical. Would you agree with that?
Yeah, absolutely. It's what is the acronym KISS? Keep it simple, stupid, for when we're talking to people about the product, and let the experts and the science just speak for itself. And if people want to read it, they can read it. It's there. but the product works when we see the results that people get.
You know, and I think I was making some notes on what you were saying there, but neuroprotective, for me as a layman, I understand what that means.
Yeah.
And I think the guy on the treadmill at the gym next to me, whether he drives a truck, teaches school, or he's a PhD in science, he knows what neuroprotective means because it's become a relatively familiar term, I would say. But if I can ask Dr. Linseman to rejoin us for a second. Keith, can you unmute Doctor for a second? Dr. Linseman, would you agree with that? Is there a further definition in neuroprotective that you'd like to add for the benefit of our sales force?
Well, I think neuroprotective, I think antioxidant, anti-inflammatory, all those come to mind when I think of immunocal. And so, you know, neuroprotection is, I don't know if it's a common public term, to be honest with you. But I think that the basically what you're saying there is that through the, you know, increasing the antioxidant pools, you're able to increase the resilience of these nerve cells, right, against stresses and different types of damage. And so that's the kind of the underlying definition of neuroprotection.
And my other question on there, Doc, for the benefit of not only myself, but everyone else listening, I remember at least 10 years ago, might be even 20 years ago, Time Magazine had on its cover for the first time about inflammation. That inflammation was linked to comment on that if you would. Inflammation and how important it is to get the inflammation down, not only for the brain, obviously, but for the rest of the body. But if you can kind of give us a Reader's Digest on that.
Yeah, I mean, an inflammation has become a very public term, right? You can't turn on the TV without seeing something about decreasing inflammation, whether it be using, you know, something to decrease brown spots on your skin. That's a type of inflammation and oxidative stress that causes that. In terms of aging, of course, inflammation increases with aging. That's why we have so many problems with our back and our knees and our brains, right? That's largely due to oxidative stress and inflammation. And so I think the interesting thing about immunocal is that the anti-inflammatory, or in my case, I work on specifically neuroinflammation, those effects really come from the antioxidant boosting effects. So inflammatory cells like microglia, astrocytes, or even certain types of white blood cells, one of the mechanisms by which they become inflammatory is they undergo mild oxidative stress. That's an underlying mechanism of inflammation within the inflammatory cell. And so probably what's happening is immunocal, through its antioxidant action by boosting glutathione, is decreasing the oxidative stress within the inflammatory cells and diminishing their ability to take on that neuroinflammatory phenotype.
Great. Well, great. So, well, Dr. Linseman, Dr. McTavish, thank you both, not only for your commitment to Immunotech and Immunocal, but also for both of you getting the educations that you have and being willing to share this stuff, you know, in public information like this with, you know, with the rest of us. So thank you both so much. And I'm going to bring on now a little bit about what's going on in the company, and that's our friend JC Martinez. And you're going to want to stick around after JC's done, because we've got two very special things to share with you today on an ongoing basis. So JC, are you with us?
I'm here, John. How are you? Thank you, Keith, for the unmuting.
I'm
Thank you
good.
very much.
Good to see you.
I'm here. You know, very, very excited for being in this spectacular and hearing Dr. McTavish and Dr. Linseman. It's great. So thank you for inviting me today. And you know, as you said, we have some updates as of today. Remember, we just heard that we have the best product, and there's no doubt about it. You know, we have the best product in the industry, of course. But we also have the best compensation plan, and we also have best promotions and best incentives. So remember, for those who are trying to go to New York, it's the last month for you to get that trip. You know, there's a lot of people that are earning that trip. So this July, it's the last chance for you to get into that trip. It's going to be amazing. It's going to be an amazing experience, you know, trying to be with more than 300 people that are going to be in New York with us. So don't take that opportunity. Don't lose that opportunity. Take that opportunity and earn the trip. Also, you know, remember that we have a lot of promotions regarding the product. We have a lot of things, you know, that are there for you. This month is the booster month, so we just heard about boosting the glutathione, as Dr. McTavish just said. So we need to boost the glutathione with booster. Immunocal booster is on sale this month, so don't lose the opportunity to take those products also. And you know, as they said, we have the best product. As I'm saying, we have the best compensation plan. We have the best company. It's the time for us to do all what it's in our hands to bring all the people to the company. It's the best time to be in this company. It's the best time to be with us, you know. Having this Zoomtacular, it's amazing because it's giving us the opportunity to know more about the product. It's good to know more about the company. And also we are doing more trainings online via Facebook every Tuesday and every Thursday. And also we have some Discovery Immunotech on Monday. Everything it's at 8 p.m. Central Time. Monday it's Discovery Immunotech, and Tuesday and Thursday are some other kind of training like the compensation plan, like the back office. So, you know, with these Zoomtaculars regarding the product, you know, regarding the leadership and also the trainings that we are offering you every week, I think you have in your hands the best business and, you know, the best personal business. So thank you very much. Thank you for being here. You know, we have more than 200 people today in this Zoomtacular. Thank you for being here. Thank you for sharing this. And remember that, you know, we are going to share this. Thanks to Keith Hooper, we are going to be able to share this and, you know, share with your people, because I am receiving a lot of calls from Mexico, from Colombia, that they are hearing these Zoomtaculars, and they are very, very excited about hearing Dr. Conchava and Dr. Gottman. Of course, today, Dr. Linseman is going to be great, and Dr. McTavish. Of course, you know, they are going to be very, very thankful for this Zoomtacular. So share it, share it. And another time, you know, you are in the best company. So share the opportunity to everyone that you can talk, share the opportunity, because Immunotech, it's here, it's for you. And, you know, we need to be very, very proud of this. So thank you, John. Back to you.
Well, thank you, JC. And, you know, you set me up perfect there, buddy, because we have two quick announcements. Because, you know, the normal question I get after these calls, like you do and like Keith does, of course, is when's the next one and who's going to be on it? Well, Mr. Molson, you want to rejoin us here and you want to share about who's going to be on with us on August the 8th? John, are you with us?
Yes, I am.
You want to share a little bit about who Dr. Nathan Lewis is?
Sure. Today we learned about neuroinflammation. Well, the same applies in a different way with regard to athletes. And we've been working for a number of years with one of the top companies in the world that uses different biomarkers measuring inflammation in athletes and helping coaches achieve that delicate balance between preventing injury, preventing illness, and achieving their top performances. This company just signed an agreement with the National Basketball Players Association, where they're going to essentially have these top players come in, get tested, and to aid their longevity on the court. And one of the chief scientists behind this company, who I've worked with over 10 years, who's worked with through 3 Olympic cycles with the British Olympic team, as a member of the English Institute of Sport, Dr. Nathan Lewis, uses Immunocal. And he's going to explain the application for Immunocal in elite athletics and explain why he uses it. So it'll be incredible insight into high performance sports and Immunocal.
You know, John, to go one step further, you know, of course, you and I have the benefit of talking on a regular basis. But I asked Dr. Linseman an interesting question yesterday about does Dr. Linseman collaborate with Dr. Lewis and with some of the other doctors that are doing our science? And of course, he gave an emphatic yes. And I know back, I believe it was in 2017, that you actually had all these guys under one roof, and men and women, by the way. It's not just male researchers. We have a lot of female researchers, obviously, as well. But John, if you can just comment on that a little bit about the collaboration, because obviously Dr. Linseman's expertise is in the head, the brain. You know, Dr. Lewis, more athletic performance, recovery, some of these other docs, different things. But if you can kind of share how you coordinate that collaboration so that the best of the best scientifically that are studying Immunocal are talking to each other so they get the benefit, obviously, of each other's research, because obviously you can't do it all alone, right? I mean, we know how dedicated these people are to normally one part of the anatomy. But if you can kind of comment on that a little bit, John, because it's such a relevant point. And it's such a point of difference about Immunocal and Immunotech, frankly, versus any other network marketing company. In my experience, there might be one or two out there. If there are, I apologize to them. But I've never seen in my 37 years a company with this level of research and scientific validation, evidence-based science, and commitment to having the best of the best study it, collaborate, talk about it. And frankly, that are not company employees. We're not paying them to say these nice things about us. They're doing it independently. So if you could just comment on that a little bit, John, how this process works.
Well, it's interesting. I mean, when we formed the company, I was an elite athlete, and I got to work with this gentleman called Dr. Bunos. And Dr. Bunos had focused primarily on the medical aspects, and I came in and I started talking about, you know, my experiences in high performance sport. And from that, Dr. Bunos really developed this holistic perspective about Immunocal and health. And, you know, I started to look at globally how Immunocal may impact us, not just with regard to a particular medical condition, but where we talk about wellness, we talk about resiliency. And what's really interesting is, you know, I tell people that essentially we've studied Immunocal from the stage 4 cancer patient to the Olympic athlete. And as Dr. Nathan Lewis will tell me, and has told me, he says, what you see in the emergency room, you also see in the elite athlete in terms of this inflammation. And, you know, what I see with regard to prehabilitation, or work on prehabilitation in cancer patients, you also see in the athlete. So it's fascinating to be at the forefront, looking at all this different research, and what you may learn with regard to the brain may apply to sports, and it's, so we organize, we have a scientific advisory board where, you know, we bring in experts who we talk to on a regular basis, get their advice on the formation of these protocols, and basically establish criteria and scientific direction. And this has been ongoing for the last 20 years. And this is, you know, all the work behind the scenes in terms of setting up what Dr. Linseman did is lots of discussions about, you know, what we decide to investigate, what are the reasons, what are the possible risks, what are the upsides, and, you know, it's you make a calculated decision. So that's.
But John, you know, as we're coming into these Zoom calls and we're doing all these, they're all connected. You know, today was about don't lose your mind, take Immunocal, and next on August the 8th, it's going to be take Immunocal so you're still able to walk. And then we're going even further after that, you know, so it's mind, body, soul, and all around good health, right?
Well, we have so much content and in-depth analysis with regard to how Immunocal acts. And this is the great asset of this company from 40 years ago, not only about how the product acts, but actually, you know, why we make the product the way we do. And it's all from insights that we've gained from doing our own research and collaborating with people like Dr. Linseman and Dr. Nathan Lewis.
Well, you know, I'm so looking forward to thank you for today. It was just amazing. And where we're going on the 8th is even.
Let me interrupt 1 second on that. Okay. So we have a lot of questions, but one is from my sister, who is a fellow cyclist. John, you will appreciate this. I think I shared this with you the other day. Now, she's got a broken shoulder, a broken elbow, and a broken hip. About 1 week out from the injury. How can Immunical help her to heal quickly?
Well, what we've seen, we haven't measured healing times in our patients. You know, we've looked at different parameters, but what we've seen with regard to our work in prehabilitation. So in prehabilitation, what you're doing essentially is you're taking a cancer patient and getting them in shape prior to surgery. And then you measure essentially how well they do post-surgery. And in essence, we come back to this word resiliency. And so we've seen this in prehabilitation, we've seen this in elite athletics, that elite athletes together with these cancer patients are subjected to insults, big insults based on all the training they do, and insults based on the surgery, the chemotherapy. So the better in shape you are and the more resilient you are, essentially the better you do. And so you would expect to see improved healing with regard to her process.
And one other question, we can't go to all the questions, obviously, for the sake of time, John, but one of the other questions that's being asked, in further Zoom-taculars down the road, can we discuss some women's issues about women's health, specific to some of the ladies' problems in aging, once again, not only in brain function, but some of the other issues as well?
Yeah, I mean, you know, when you put together a scientific strategy, you have to study the things that you're pretty convinced will work and that you understand. And so, you know, we have a broad array of things we've studied, and then there's a very disciplined process in terms of why we study that. And so there's other areas that, you know, we haven't studied that possibly we could look at those questions in the future. But, you know, with regard to women's issues, these are not things that, you know, we've measured. For instance, in cancer patients or cancer studies, you're looking at quality of life parameters. So, you know, for instance, are you able to put weight back on the cancer patient? So what I'm presenting here and what I'm asking to be presented are things that we've studied over the course of 40 years. And, you know, just like anything else, you can't study everything. So you make a very disciplined decision about things you do study.
Exactly. Well, all I can say is this, you know, this has been an amazing day. That, you know, Dr. Linseman was amazing. And, you know, once again, John, thank you for all that you do, have done for all these many 2.5 decades that we've been together on this project. And, you know, I'll just make a comment and I'll throw it back to Keith, John, and like you to comment as well. We have one other quick thing to do after that. And it's just this, you know, when we started this, when I walked in the office there in Vadroy back in 1997, and you and I met the first time that day, and you introduced me to Dr. Bunos, I understood the product from the standpoint of people who were sick at that point. The research, you know, a lot of the research was on that, but to think how far we've come, to see the research that Dr. Linseman, for example, is doing, and some of the other research are doing, or the athletic performance. I mean, we've gone such a long way in understanding what Immunical does, and obviously the role of glutathione in the body and everything else. I mean, I remember when we started this, we had to say to people, glue the tie on, because nobody ever heard of glutathione outside of medical researchers. I'm not even sure MDs had heard of it at that point. But nonetheless, you know, here we are 25 years later, and you know, it's become something that's being talked about, you know, on public stages by the Dr. Oz's and some of the, you know, the commercial docs, if you will, and on and on and on, how much glutathione pops up. But to think how far we've come, and that array, like you said a minute ago, from the stage 4 cancer patient, maybe at the end of their life, perhaps, to the world-class athlete competing, you know, in the Olympics or professional athletics. I mean, and everyone in between. That's what's so amazing, is the scope and magnitude that Immunical is truly for everyone. So I think it's been an amazing thing. It's been an amazing day. But let me turn it over to Keith for a minute there. And then, John, if we can have a couple quick comments from you, and then we have one other thing to post, and we'll be done. Keith?
And absolutely, what an amazing day. And Dr. Linseman, thank you so much for being here. In fact, I got a question for you that has been posted here, and I'm actually having a conversation with a gentleman on Tuesday. With what you're seeing with Immunical and what it's done, what would how do you think, how beneficial would Immunical be for someone that's trying to detoxify from drug addiction?
That's a tough question.
We didn't expect an easy one from me, right?
Yeah, it's a little outside my area of expertise, but certainly glutathione is involved in conjugating a lot of metabolites and things that tend to be elevated during detoxification. And so it could be beneficial in that regard because it's involved in, obviously this is more a liver question because it's metabolism really, I think is what you're looking at. But I would imagine that elevating glutathione would help form a lot of glutathione adducts, which would help clear the body of some of the products, the metabolites and that you have that you're trying to get rid of during that detox process.
Well, thank you so much for that. And so, John, Mr. Molson, that prepares you for my call I'm going to have to call you on Tuesday for regarding that issue with a friend of mine. So, once again, Immunical is one of those products that's such a broad spectrum. So often people say, well, what will this product do? And we get in, like we're looking at the chat here, Mr. Solider, and a lot of specific questions. The reality, Immunical works every time given time, and it works, it just works, and challenge it. So, so Mr. Molson, thank you so much. I know you've got another comment for us, and then we've got just a couple things to share with folks regarding what's coming up.
Well, I'll let people know that the last speaker we're going to have is a gentleman here at the University de Montréal who ran our aging study, and this protocol was designed by Dr. Drobe. And so part of healthy aging is related to good muscular strength and muscle function, and that's what we measured in this study. And so one of the researchers will present on that call. So that's another thing to look forward to.
It's going to be exciting, and when once again, and what we're doing here on the 8th, and I'm going to share that real quick. We've got Julie coming on with us there, John, because we really want to share all of the fact of using Immunical gives you that advantage, and it's physical, emotional, all around good health, right?
I want to finish off with one important content. In this study, we selected from 65-year-olds to 80-year-olds. And it was interesting because in that study, roughly 70 to 75% of the participants were women. So John asked me the question if we have any women's issues. And I asked the researcher, why were women a higher proportion? He says, because women tend to take better care of themselves and think about these things. And so that's why we have higher enrollment of women in that study. So we'll talk about that when we discuss the aging study.
Great.
Okay. Well, thank you for that. And it's exciting what we're doing on these Zoom calls. And thank you so much, Mr. Molson. Thank you, Dr. Linseman, for being with us. I mean, you delivered. I told people to be here, and some of them was pretty strong on why they needed to be, and I'm sure they were not disappointed today. So thank you so much for giving us your time here. So Mr. Solider, we've had a very, very powerful day, and we've got some more things to share with people that's coming up. I know we want to touch on a couple things here regarding the book. Once again, about sharing this, because the idea is to share the product, share this business opportunity with people, and it comes from knowledge. So you can get this book that John co-wrote, and or John wrote and allowed me to be on. My name is real small there because I didn't have to do very much. John did all the work. But get over to tremendousleadership.com, Tremendous Leadership, and you can pick that book up there off of there. It's also available on Amazon. But get it over to Tremendous Leadership and get tied into all the things we do here. Get over to XYZTools.com. That's www.xyztools.com. That's how we communicate with everybody to let them know about these events and all the things that we have going on. So, Mr. Soller, it's been a pretty spectacular day.
I'll tell you what, we apologize for the one brief interlude of people who obviously really needed to listen to Dr. Linseman maybe more than we all did because their brains aren't functioning properly. They certainly need Immunical. So we apologize for that. Sometimes these Zoom hackers, it's sad because they could be doing a lot more with their lives. Let's put it that way. I'm sure we're all in agreement. So apologize certainly for that happening, but fortunately it only happened for about a minute. So we caught it, but it's been a great day. And you know, we are a different company and you know, to have people like Dr. Linseman, independent, if Immunotech was or was not a network marketing business, to have a world-class scientist like Dr. Linseman working with our product. And, you know, we'll add Dr. Lewis in there, we'll add the other doctors in there, to have these people where they don't care how we sell this stuff. I mean, they care that we sell it obviously in an ethical and legal manner, I'm sure, but that's not their concern. When they go to work every day, their commitment is to the excellence that this product can help to deliver for humanity. And once again, you know, that's a point of difference with Immunotech research versus, frankly, most of what's out there in the marketplace. Not only network marketing, by the way, but most nutritional supplements. So anyway, I'll just leave it with that. We'll talk to everybody August the 8th, and it's going to be another Zoom-tacular day. It's going to be phenomenal again. And you know, once again, thank you, Dr. Linseman. Thank you, Dr. McTavish. Thank you, Mauricio. Thank you, John Molson, as always. Thank you, JC. And most importantly, thank you to each and every one of you who took time on your Saturdays to listen to these calls, to learn more, to share more. You know, these calls are for you to benefit from. We're still learning, 24 years doing this. I'm still learning things. As I said, I had 4 pages yesterday from Dr. Linseman. I have 8 more today. And he went over the same stuff. So, you know, it shows why repetition is the mother of invention, so to speak. So I know I learned a lot today. I hope you did too. So John Molson, thank you again. Keith, thank you again. And we'll talk to everybody here in, bye for now.
Have a great day, everybody.